All right, so this is nervous system or neural tissue rather, part 2. And I'm just switching my computer over into drawing mode. So where we left off, I said, let's just go ahead and save neuroglia for next time. And one of the reasons for that is that they are a complex set of cells by themselves. And that means that I just thought that they kind of deserve their own deal. So that's what we're doing here. So without further ado, let's get into these special cells. So the first two types, ependymal cells, and then I'm trying to get to the next slide, astrocytes, so a few types rather. The first ones, and I'll go back and read you these and talk about them, but we're going to start with CNS, central nervous system, glial cells, because although there's glial cells throughout the entire nervous system, the type of glial cell varies between the central nervous system and the peripheral nervous system. So we don't have the same cells depending on if we're in the CNS or PNS. So from that perspective, if I show you a picture and you see, for example, that the picture contains these cells, oligodendrocytes, which we'll talk about in a little bit, you would conclude from that that you've got to be in the central nervous system because oligodendrocytes do not exist in the peripheral nervous system. So it's just an example. Okay, so these cells are going to line ependymal cells specifically. Some people pronounce them epidymal cells with the emphasis on the di, but ependymal cells is more common. Again, doesn't matter how you say it as long as you can spell it correctly. But regardless, these are going to line the fluid-filled spaces in the central nervous system. So in the spinal cord, that is the central canal, which is a tube that runs through the middle of the spinal cord. And in the brain, that's going to be the ventricles, which you don't really know about yet, but these are spaces filled with fluid. So you'll learn all about those when we get to the brain stuff. So the ependymal cells or ependymal cells secrete cerebrospinal fluid. And I'm going to put an asterisk on this because cerebrospinal fluid is really sort of modified blood plasma. And so to say that the ependymal cells secrete it all by themselves is kind of not true. All of the blood filtrates in your body, including your cerebrospinal fluid, ultimately come out of pressurized capillaries. And so in the brain, those pressurized capillaries are part of something called a choroid plexus, which again, I'll show you later. But basically, fluid leaks out of them, and then that fluid is modified by the ependymal cells and some other glial cells that we'll discuss. So I just don't want you to see the word secrete and think that they're doing it all by themselves, because that's not the case. So these are also going to monitor the composition of cerebrospinal fluid. So they participate in the blood-brain barrier and also ions, pH, glucose. So just making sure that the right stuff is in the cerebrospinal fluid and that anything that shouldn't be there isn't. And because they are ciliated, they can wave their cilia around and circulate the CSF within the ventricles. They participate in propelling the cerebrospinal fluid's movement. Because you don't just have the same juice all the time. The cerebrospinal fluid is constantly being synthesized, circulated, and then reabsorbed. More on that later. Okay, astrocytes. So astro reflects star. And if we just give them a look, what we're going to see is they have multiple extensions and projections. So here's one, which gives them a sort of stellate or star-like appearance. So that's why they're named that. So these help to maintain the blood-brain barrier, as you can see over here, and I'm going to switch away from this yellow color because it's hard to see the yellow. But this astrocyte is sending little projections, and then that results in a bunch of little feet that wrap around this capillary, and they control what is able to leak out of the capillary. So that's what we call our blood-brain barrier. So those feet are going to control what's able to get through, because there's certain things that are damaging to our brain and we don't want them anywhere near our central nervous system. Instead, we're going to keep them out and only take the stuff out of the plasma of the blood that is good for us and good for our neurons. They also provide structural stability. So you can see that they are bracing the axon of this neuron and this capillary away from each other. So they provide basically a framework or a scaffold almost for the central nervous system. In doing so, they're able to stabilize the area where neural tissue is damaged and can provide a place where, let's say you get a damaged axon, they can stabilize the broken end of the axon until it grows a new extension. So one example. And by their work with the blood-brain barrier and their control of what gets across, basically they're just going to regulate the concentration of things like nutrients, amino acids, glucose, ions. So these are cations. And also respiratory gases like carbon dioxide and oxygen. So they have a huge role in the health and well-being of your central nervous system. So although neurons are kind of the stars of the show and neurons get the most attention, etc., so forth, it's impossible to have a functioning brain without astrocytes. So, and the other glial cells as well. So they're all part of this ecosystem that allows our neurons to do the work they do. By squeezing or relaxing the capillaries, they can regulate blood flow. And if you want to look at this, you can get a functional MRI, which shows you in real time where in the brain is receiving the most blood. And that can provide researchers with valuable information about what areas of the brain certain thoughts are processed in, or feelings, or what have you, because they're going to demand more oxygen. And finally, they can enhance synaptic function via their association with axons. But we're not going to get too much into that because just kind of beyond the scope of this class. However, what I will say about it is this. They are involved in the reabsorption and recycling of neurotransmitters at some synapses. But again, exactly how they do that, you don't need to know. So, moving on to microglia. Micro means very small. So these are actually related to macrophages. They're same stem cells, and because they come from the same stem cells, that means that they have related functions. So one of the things that these little guys do is they eat up debris. So as your neurons function, they're going to produce wastes and debris. So misfolded or broken chunks of neurotransmitter. They can also eat pathogens like viruses and bacteria, although the brain environment is pretty tightly controlled so that viruses and bacteria shouldn't really be in there, but if they show up, microglia will deal with them. But regardless, microglia play a critical role in basically resetting your brain, and that specifically happens mostly while you're sleeping. So here's how this works. All that debris and waste and those pathogens, if they're present, are going to be floating in the cerebrospinal fluid, which is the interstitial fluid of the central nervous system. And that means that they can do things like accumulate in the wrong place and block synapses and make your cognition poor and all kinds of other bad stuff. So, when you go to sleep and you're entering the successive cycles of dropping into deep sleep and then jumping back up into REM sleep, we'll talk more about the sleep cycles later in this term, but they're very interesting. One thing that happens is the rate at which your cerebrospinal fluid is circulated. Oops, I got a little ahead of myself. CSF. The rate of the circulation of CSF dramatically increases. So what that's going to do is move all of that debris and waste and crud around from its place where it's accumulated. It's going to sweep it up in the current and carry it around. And that gives microglia the opportunity to eat it up. So microglia are going to eat the waste. They encounter as a result of this. So it's a team effort. If you want to get rid of the junk and waste and basically neuron poop that's accumulating in your brain as you use your brain during the day, you have to go to sleep and you have to drop into deep sleep and go through some REM cycles. And during that time, the process that I just explained happens. And that's why when you wake up, you feel refreshed, hopefully. And that's also why if you don't sleep or don't get good quality sleep, your cognition suffers. So this is me telling you, if you feel like you need to pull an all-nighter to study or you're losing sleep about class and such, prioritize your sleep. Be protective of it because it's really important for good cognition. And if you're going to learn the huge, huge amount of information for this class, you need to have good cognition. So make sure you're sleeping and do your best to sleep well. Okay, oligodendrocytes. So oligo means many. Dendro means tree or branch. Really, it means tree. Branch is ramus, and then cyte is cell. So these are called oligodendrocytes because they have many extensions, kind of like a tree. And what they do is they wrap those extensions around axons, forming the myelin, which is a fatty covering that helps to speed up the transmission of action potentials. And I'll describe how it does that also. But essentially, without myelin, your speed of neural transmission would go a lot slower. So these provide structural organization as well. So they do the same kind of bracing function that the astrocytes do, in addition to their myelination. So let's talk a little bit about myelinating just to begin with, and we'll sort of build up to describing how it functions in action potentials because it does matter a lot. So if we look right here, you can see that the little foot is wrapping around and around and around and around this axon like a jelly roll. So this functions in the same role as electrical insulation of wires. So if you were to go to your house and pick an electronic that's plugged in and cut the wire of it, or you cut a fiber optic cable that carries information in the form of electricity, what you're going to find is that inside of the wire, there are smaller wires with various wrappings, and then they're all wrapped in one big wrapping. And that electrical insulation prevents the electrons that are cruising through those wires. It prevents them from leaking out sideways or getting on the wrong track, etc. So that's what electrical insulators do, more or less, and myelin provides the same function for neurons. So it increases the rate of impulse conduction along what's called the axolemma, which is the cell membrane of the axon. So this is similar to sarcolemma, which was the cell membrane of a skeletal muscle cell. So lemma refers to that membrane, and then whatever comes first is telling you what it's the membrane of. So axolemma is the membrane of the axon itself. And I have a meeting to go to, so I'm going to pause here and pick up when I get back from the meeting. So I will see you in a little bit. Or it'll be no time at all for you. See you in 2 seconds.